By the time most people reach 70, they have lost more than a third of the bacterial species that were present in their gut at 40. Not weakened, not temporarily reduced, lost, absent from their internal ecosystem in a shift so gradual and so consistently silent that no symptom announced it, no standard test detected it, and no doctor thought to mention it. What is dying is not random. The bacteria that age and modern life push toward extinction first are not the marginal ones, not the species whose absence goes unnoticed. They are the species that were doing the most consequential work. The species producing the compounds that maintain the integrity of your intestinal wall. The species manufacturing the molecules that crossed into your brain and suppressed the inflammation that ages neurons. The species whose population collapse, confirmed now across multiple research cohorts, is associated not merely with digestive changes, but with a measurable acceleration in the rate at which the brain grows older. Something specific is dying in your gut. And once you understand what it is, what its name is, what it was protecting, and why the supplement industry has never told you about it, the path to addressing what is happening becomes available to you in a way that it has not been before. In this video, I am going to name the 3 species that decline most consistently and most consequentially after 65, tell you exactly what each one was doing for your brain, and give you the specific intervention for each that the research supports. Not general gut health advice, species-specific, precise enough to be actionable this week. At around the 9-minute mark, I need to tell you about a bacterial species, the most abundant beneficial organism in the healthy adult human gut, that has never appeared in any probiotic supplement you have seen on a pharmacy shelf. Not because the manufacturers haven't tried, because it cannot be kept alive through the manufacturing, packaging, and shelf life process that supplements require. The supplement industry cannot sell it to you, which means no one in that industry has any financial reason to tell you it exists. I will tell you its name, what it does for your brain, and the one food that feeds it. I have spent more than 3 decades in geriatric medicine, and the research on specific gut species and brain aging has moved faster in the last 5 years than I would have predicted. What has emerged is not a general picture of gut health matters. That picture was already clear. What has emerged is a precise one. Specific species, specific functions, specific consequences of specific losses. Let me give you the frame before I give you the science. In the late 20th century, ecologists studying Yellowstone National Park after the reintroduction of wolves documented something that had not been anticipated in its full scope. The wolves did not simply reduce the elk population. Their presence changed the behavior of the elk, which changed where and how the elk grazed, which allowed vegetation to recover in specific areas, which stabilized the riverbanks, which changed the course of the rivers. One species, reintroduced, altered an entire landscape through a chain of effects no single intervention was expected to produce. The scientists named this phenomenon trophic cascade, and the species at the center of it, the one whose presence or absence restructures the entire ecosystem, they called a keystone species. Your gut microbiome has keystone species, and after 65, those keystone species begin to disappear. Not every bacterial decline matters equally. Not every loss has equal downstream consequences. What makes the species I am going to name today different from the general picture of age-related microbiome decline is that their loss does not simply reduce microbial diversity. It removes the scaffolding that everything else depended on. And what follows is not a gradual proportional reduction in gut function. It is a cascade. The first species is one most people have never heard spoken aloud, including by their physicians. Its name is Faecalibacterium prausnitzii. In a healthy adult gut, it is typically the single most abundant beneficial bacterial species present, comprising between 5 and 15% of the total gut bacterial population by some estimates. It is, by the measure of presence, the dominant force in a healthy microbiome, and it is among the first to collapse with age. Faecalibacterium prausnitzii is the gut's most prolific producer of butyrate. The short-chain fatty acid I have described in other videos as the compound that maintains the intestinal wall, suppresses systemic inflammation, crosses the blood-brain barrier, and directly protects neurons. But F. prausnitzii does something beyond butyrate production. It actively suppresses a specific set of inflammatory signaling molecules, including IL-8 and IL-12, in ways that reduce intestinal and systemic inflammation independently of butyrate. It is an anti-inflammatory organism in itself, not merely a factory for anti-inflammatory molecules. Multiple research groups have now found an inverse relationship between F. prausnitzii abundance and cognitive performance. Lower F. prausnitzii, worse cognitive outcomes. The association has been replicated across cohorts of different demographics and geographies. In people with Alzheimer's disease, F. prausnitzii levels are consistently and significantly reduced compared to age-matched healthy controls. The second species is Akkermansia muciniphila. If F. prausnitzii is the gut's anti-inflammatory engine, Akkermansia is its wall builder. Akkermansia lives in the mucus layer of the intestinal wall, the protective coating between the gut's bacterial contents and the intestinal lining itself, and its primary function is maintaining that layer. It stimulates the goblet cells of the intestine to produce mucus, feeds on that mucus in a cycle that keeps the production system active, and in doing so, maintains the physical barrier that prevents bacterial toxins from crossing into the bloodstream. When Akkermansia declines, which it does with age, with antibiotic use, and with low-fiber diets, the mucus layer thins, the intestinal wall becomes more permeable. The lipopolysaccharides that I have described in previous videos begin to cross, and the systemic and neurological inflammation that follows is the direct consequence of losing the species responsible for keeping the wall intact. In research comparing gut microbiome profiles of older adults with and without cognitive impairment, Akkermansia is consistently lower in those with impairment. In animal studies, restoring Akkermansia has been shown to reduce amyloid beta burden and neuroinflammation. The translation to human intervention is an active area of research, but the directionality is consistent and the mechanism is established. The third declining species is not a single organism but a family, the Bifidobacterium genus. Bifidobacterium species are among the most extensively studied gut bacteria in human health, and their relevance to brain aging runs through a pathway that is almost never discussed outside of specialized research circles. Bifidobacterium species produce B vitamins. Specifically, several members of the Bifidobacterium genus are significant producers of B12, cobalamin, and folate. Both of these vitamins are critical for neurological function. B12 is required for the maintenance of the myelin sheath, the protective coating around nerve fibers, and for the synthesis of neurotransmitters including dopamine and serotonin. B12 deficiency produces neurological symptoms that overlap with cognitive decline, memory impairment, processing slowing, mood changes. After 65, Bifidobacterium abundance declines significantly, and with it, the gut's contribution to B vitamin synthesis declines. B12 deficiency in older adults is extremely common. Estimates suggest it affects between 10 and 30% of people over 65. Most of those individuals are either supplementing B12 or have been told their levels are low without a clear explanation for why. The explanation, in many cases, is the bacterial species that used to produce it. I want to tell you about a patient I worked with, no name, because this pattern is representative of something I have encountered multiple times, not an isolated case. This patient had been supplementing B12 on their physician's recommendation for just over 2 years. Their levels had initially been flagged as low at a routine blood draw. The supplement had been prescribed. At each subsequent annual appointment, the levels had risen modestly but had not normalized fully, and the supplement had been continued. What nobody had asked at any of those appointments was why the levels had fallen in the first place. When I asked that question and we looked at the clinical picture in full, the picture that emerged was of a gut microbiome that had been significantly altered over several years by a combination of 2 antibiotic courses and long-term proton pump inhibitor use. The Bifidobacterium populations, in particular, were severely depleted. The gut had stopped producing the B vitamins it had previously supplied. The supplementation was replacing a function that had disappeared. but the reason for the disappearance had never been named. And with no one naming it, the possibility of addressing it, of restoring the Bifidobacterium populations and supporting a recovery of the gut's B12 synthesizing capacity, had simply not been part of the conversation. That patient, with appropriate support for gut microbiome restoration, increased prebiotic intake, targeted fermented food consumption, the removal of unnecessary factors suppressing the gut environment, showed improvement in Bifidobacterium abundance on a follow-up stool analysis over 6 months, with a corresponding improvement in B12 levels that eventually allowed the supplement dose to be reduced. Their physician was genuinely surprised. They were not aware that the B12 deficiency had a bacterial source. Now, the 9-minute mark, the species I have been building toward. Faecalibacterium prausnitzii, the dominant beneficial species in the healthy gut, the primary butyrate producer, the anti-inflammatory keystone species I described at the start, cannot be purchased in any supplement. The reason is biological. F. prausnitzii is what researchers call a strict anaerobe. It cannot survive exposure to oxygen. The manufacturing processes required to produce, encapsulate, package, and distribute a probiotic supplement expose the bacterial culture to conditions that kill this species before it ever reaches a shelf. Companies have attempted to develop oxygen-stable formulations. To date, no commercially viable probiotic containing live F. prausnitzii exists for consumer purchase. This means that the supplement industry, which sells billions of dollars of probiotic products annually, has no product that addresses the species most consistently linked to brain aging through the gut. No probiotic capsule, no fermented supplement, no high-potency multi-strain formula. The species is absent from the market because the market cannot manufacture it. What does feed Faecalibacterium prausnitzii is equally specific. F. prausnitzii is selectively fed by inulin-type fructans, a category of prebiotic fiber found in highest concentration in a small number of common foods. Jerusalem artichokes are the richest natural source. Leeks are the most consistently available. Garlic, asparagus, and chicory root are also significant sources. These foods, consumed regularly, produce the conditions in the gut that allow F. prausnitzii to thrive. The reason most people do not eat these foods in abundance, and this is worth noting, is that in a gut with already depleted F. prausnitzii populations, inulin-type fructans initially cause significant gas and bloating, not because the foods are harmful, because the bacteria that would otherwise ferment them efficiently are absent, and the fermentation is initially chaotic. Many people try these foods, experience the gas, and conclude the foods disagree with them. The opposite is true. The gas is the indication that the gut needs them most. For Akkermansia, the research points to pomegranate polyphenols, L-tannins in pomegranate, as among the most specific natural stimulants of Akkermansia growth. A small glass of pomegranate juice daily, or a concentrated pomegranate extract, has been used in research protocols specifically targeting Akkermansia restoration. Fasting intervals also support Akkermansia. This species feeds on the gut's own mucus layer during periods without food, which paradoxically stimulates mucus production and gut wall maintenance. For Bifidobacterium, galacto-oligosaccharides, found primarily in legumes and available as a prebiotic supplement, are among the most specifically studied Bifidobacterium feeders. Combined with consistent fermented food intake, they support the restoration of B vitamin-producing populations in a way that generic fiber intake alone does not. Your gut microbiome is an ecosystem, and ecosystems can be restored. The principle that allowed wolves reintroduced to Yellowstone to stabilize rivers and reshape a landscape applies to the living environment inside your gut. Restore the keystone species, provide what they need, remove what suppresses them, and the cascade runs in the other direction. The wall strengthens, the inflammation reduces, the compounds that protect neurons begin to flow again. This is not a metaphor. It is the biological reality of what the research now shows. And it begins with knowing which species to restore and what those species eat. 3 things. Specific enough to start this week. The first is the Keystone Species Diet Protocol. Introduce gradually and consistently the foods that feed the 3 declining species. For F. prausnitzii, 1 to 2 tablespoons of leek or 1 small Jerusalem artichoke, 3 to 4 times per week, building slowly to daily if no significant digestive discomfort prevents it. For Akkermansia, a small glass of pomegranate juice or 2 to 3 tablespoons of pomegranate seeds daily. For Bifidobacterium, 1 cup of cooked legumes, lentils, chickpeas, black beans, 3 or more times per week, combined with 1 daily serving of fermented food with live cultures. Begin each addition separately across different weeks to allow the gut environment to adjust. The second is the stool microbiome analysis conversation. Ask your physician or gastroenterologist about a comprehensive stool microbiome analysis. Tests marketed under names including GI-MAP, Viome, or Geneva Diagnostics Comprehensive Stool Profile. These tests measure the abundance of specific bacterial species, including F. prausnitzii and Akkermansia, directly. If your levels of these keystone species are critically depleted, the test transforms the conversation from general gut health advice into specific, measurable intervention. Coverage varies, but these tests are available and increasingly ordered by integrative and functional medicine practitioners. The third is the B12 source investigation. If you have been supplementing B12 or have been told your B12 is low, ask your physician, is there evidence that restoring Bifidobacterium species in my gut could improve my B12 status? Could gut microbiome restoration be part of the management plan alongside supplementation? This question is clinically legitimate, is supported by research, and opens a conversation most B12 supplementing older adults have never had. Addressing the bacterial source rather than only replacing the vitamin is a different, and potentially more durable, approach. Go back to where this video began. By the time most people reach 70, more than a third of the bacterial species that were present in their gut at 40 are gone. And what has gone is not random. It is the keystone species, the wall builder, the butyrate factory, the B vitamin manufacturer. The organisms whose absence does not simply reduce function, it cascades, it destabilizes, it accelerates aging in the organ that cannot afford to age any faster. You now know what is dying. You know its name. You know what it was protecting. And you know what feeds it. If someone you know is being told their low B12 or their cognitive changes or their persistent gut discomfort is simply part of getting older, please share this video with them, because the species at the center of those connections has a name, and named things can be addressed. Before next week, I want you to try one specific thing. Tonight, not tomorrow, tonight, include 1 tablespoon of finely sliced leek in something you eat. Soup, eggs, a salad, anything. 1 tablespoon of leek. The reason I am asking specifically for leek is that it contains one of the highest concentrations of inulin-type fructans of any commonly available food, the specific prebiotic fiber that Faecalibacterium prausnitzii selectively ferments. If you notice any gas or bloating in the 2 to 3 hours that follow, write it down, because that gas is not a problem. In most cases, it is the sound of a depleted bacterial population beginning to ferment something it has not had adequate food supply for. It is a signal. And next week I am going to explain what that signal specifically tells you about the current state of F. prausnitzii in your gut, and what to do differently depending on whether the gas is present or absent. Come back with what you noticed. One important note before you go. Everything in this video is for educational purposes only, and is not a substitute for the guidance of your personal physician or a registered dietitian. The dietary changes described, including inulin-containing foods, may cause digestive discomfort, particularly in people with irritable bowel syndrome or other gastrointestinal conditions. Please introduce any new food gradually and discuss significant dietary changes with your healthcare provider. The stool microbiome tests mentioned are available but vary in clinical validation. Discuss with your physician whether they are appropriate for your individual situation. The goal of this video is to give you a more specific picture of what the research shows and the questions to bring to your next healthcare conversation.